Scott Harper, Principal Investigator at the Center for Gene Therapy at Nationwide Children's Hospital. And he's going to talk about supporting the translation of the CMT1A gene therapy through AAV-mediated silencing of PMP22 in mice and non-human primates.
Thank you. Thanks to the organizers and to CMTRF for inviting me here to speak. It's a real pleasure to be here. I'm going to tell you a little bit about gene therapy again. I think we've heard a lot about it and so had some great talks, but I think repetition is a good thing. So here are my financial disclosures. The most noteworthy one is that I'm a co-founder and currently serve, in addition to my academic position, as Chief Scientific Advisor for Armatus Bio. And before I get started on the talk, I want to recognize the team here. This is, Cleopas already referred to this study. This is work that we've been doing in strong collaboration with Cleopas and Marina, a very talented PhD student who then became a postdoc in Cleopas's lab. So this work is really, a strong collaboration between our lab and his lab. But also, and I want to recognize in my lab as well, Lindsay Wallace, who's sitting over here, and Merlin Thangaraj, who's a postdoc in the lab, who's done a lot of work helping us phenotype some of the non-human primates that are I'm going to show you in this study. And then finally, I want to point out that Armatus Bio has funded this. This is a company we co-founded, and in particular, Rachel Salzman and Brian Price have been instrumental in helping us move this forward. So I'd like to recognize them as well. And then finally, we did all this work at Amplify Bio, which is a division of Battelle, a nonprofit based out of Columbus, Ohio. So our goal today is to develop, the goal I'm going to tell you about for this project is to develop a gene therapy for CMT1A. And so our cell target is Schwann cells. We're targeting abnormally high PMP22 levels, and our goal is to reduce those to healthy levels. And our approach is to use something called RNA interference. Probably some of you in the audience are familiar with this. I'm going to tell you a little bit more about it. And our delivery vehicle is adeno-associated virus, specifically serotype 9. Now, I want to point out here, too, that so Dr. Boneman gave a beautiful talk earlier today. He's described the trial for giant axonal neuropathy using AAV9. Zolgensma is approved. It's treating patients with SMA using AAV9, and Cleopas just showed you a whole list of gene therapies in the preclinical stage that are using the same vector. So we are privileged to have in the audience here, and going to be giving a talk, the person who invented that or discovered it, Guanping Gao. He's somewhere here. Maybe he just walked out. Okay, there he is. And so I think we all owe a debt of gratitude to Dr. Gao, too, from the University of Massachusetts for discovering this vector that all of us are using and has given rise to these clinical products. So this is just, we've talked a lot about CMT1A. The major point I want to make on this slide is that we have these elevated levels of PMP22, about 150% that we're trying to reduce down to healthy levels. And our approach is to use gene therapy. So, we all know what, I think we all have heard about gene therapy. We sort of think we know what it is, but I've asked students at Ohio State where I teach, how to define it. And sometimes people stumble on what gene therapy actually is, how to define it. So the simplest way in my mind is this: treating disease with nucleic acids, meaning DNA or RNA. And, the concept of gene therapy has been around for a long time, many decades. There's a paper from 1972 published in Science postulating that gene therapy might be possible. And now it's no longer science fiction. We have approved products on the market using a number of different viral vectors. The major points about gene therapy that I want to emphasize here, though, is that what we intend in using gene therapy is that it's a one-time treatment, if it works well. Adeno-associated viruses that we're using in this study and other studies are stable in post-mitotic cells. So we hope that if we can stabilize a disease in a cell that's not dividing, that genome, that therapeutic genome that the virus is delivering should last essentially forever. We don't know if forever is true, but it should be many, many years. I think the longest anybody's looked so far in a human is about 10 years, and they're still there after one treatment. Let's move on to the next one then. So we've talked about AAV vectors. We're using AAV9, like I said, and essentially what we're doing is we're cloning a microRNA, and I'll tell you about that in the next couple slides, inside an AAV genome. So we're actually cloning a piece of DNA that encodes a therapeutic microRNA that we've designed to knock down PMP22. And then we package that inside an AAV vector and deliver that to mice. And then I'll show you some data in monkeys as well. So we've heard a lot about gene replacement. So when people think about gene therapy, I think the first impression is, well, we're talking about gene replacement therapy, right? So there's a gene that's missing and we want to add it back. That's sort of traditionally been where the field is. But we don't want to replace, we don't want to add more PMP22 as a therapy. We want to be able to reduce PMP22. And so our approach is to use something called RNA interference. This was discovered in worms back in 1998 in a famous Nature paper. And a few years later, it was shown that it was actually all eukaryotic organisms have it, including humans. And that sort of changed the game for gene therapy. No longer were we just limited to doing gene replacement. We could actually knock down genes as well and reduce their expression. So what is RNA interference? It's a natural mechanism of gene silencing. And so all of us sitting in this room have genes, microRNAs, and their genes. We have about 1,800 in our genomes. And they encode these hairpin structures that are shown here on the slide. These hairpin structures don't code for protein. They instead are processed by the cell in a certain way to give rise to a very small 22 nucleotide functional component called we call it a guide RNA or a guide strand. And that's shown here, underlined. Oops. Underlined. That's the functional component of this. And the idea here is that these natural microRNAs that we all have, we can essentially switch out the guts of the molecule, this underlined part, and retarget these to whatever disease gene that we want to knock down. So in our case, for this study, we want to knock down PMP22. So we have changed the sequences of the microRNA so we can redirect this sequence to PMP22. And that by doing so and expressing this inside the cell with an AAV vector, we can find PMP22 and direct it to natural cellular gene silencing pathways to reduce its expression. So the idea here is it's sort of like finding a needle in a haystack using base pairing of nucleic acids. So the study I'm going to tell you about now, I'm finally getting to the data, is we're using RNA interference. I told you we're using AAV vectors. We're trying to target PMP22, and our approach is to deliver using intrathecal gene therapy. So it's similar to what Dr. Boneman talked about, similar to some of the work that Cleopas just talked about as well. And again, we intend this to be a one-time treatment. The idea is that we, again, just to remind you, clone our expression construct as an AAV genome inside the AAV capsid using AAV9. We deliver that to cells, and we hope that we are hitting Schwann cells, and we actually do. The virus then gets inside the nucleus, it delivers the genome, which can then express our microRNA, and that microRNA gets processed and then eventually causes knockdown of PMP22 and reduction of protein. So we published a paper, Cleopas's lab and our lab published a paper in JCI a couple of years ago, describing the first proof of concept for this in mice using the C61 mouse, which is a nice model for CMT1A because it expresses about 1.5 times of normal PMP22. So it's relevant to the human disease, but it also has 2 mouse copies. So it's expressing overexpressing a human transgene on the background of a mouse. So it has 2 copies of the mouse PMP22 as well. And so, when we started to think about how to design the microRNAs to begin with, we wanted to make sure that we could translate anything we were testing in a mouse to a human. And so from the get-go, we designed our constructs so that they could knock down both human and mouse PMP22 equally. And that way, if we found any beneficial effects in a mouse, we could translate that then to humans. So that was an important feature. We did some initial screening in vitro in a cell line, and this is from our paper. And we identified one sequence that we call 871. Now this is a microRNA that can equally bind mouse and human PMP22 in the 3' untranslated region of the transcript and knock down both genes. And another reason this is important is because I think probably this room knows if you have too much knockdown, you can develop HNPP. So we wanted to make sure that we're not just capping our knockdown at wild-type mouse levels. We want to be able to make sure that we could start at 150%, reduce it to healthy levels, and be confident that we're not inducing HNPP. So again, I mentioned that we published some proof of concept in the mouse, and this paper, I'm not going to go into the mouse data too much. This is just the one slide I have on it. This paper's published. We've been doing some more data or some more dose finding studies to help support translation with the support of Armatus Bio. So some of this data is published and some of this data is new. I'll just point you over here that the average knockdown we're getting through our dose response is anywhere from about 30 to 60% in the mouse in Schwann cells of PMP22. And that's showing benefit, behavioral benefit, knockdown, normalization of protein levels. I would encourage you, if you're interested, to look at this paper. We have another one coming soon on this study. But the point is, we've done this in a mouse now. How do we scale this up? And, I'm a molecular biologist. I wouldn't consider, I would say I dabble in neuroscience. And so I had a lot of people that I know and respect who know nerve anatomy. They understand Schwann cell biology, and they were convinced that it was going to be very difficult for us to deliver an AAV9 vector intrathecally to a large animal model and expect to be able to hit the distal part of the nerves. And, and I understand the idea here, we're in a mouse, say, for example, the sciatic nerve of a mouse is only about 2 centimeters, right? So if we do an intrathecal delivery in the lumbar region of the spinal cord, it only has to travel a certain distance. But in a human, maybe up to a meter. And it was unknown whether or not that could actually, this vector could do that job. So I have to also just point out that Armatus Bio was brave to sponsor this because it was an expensive study. We didn't know what we were going to find, and a lot of experts were saying this isn't going to work. But we went ahead and did a non-human primate study anyway because we needed the data. CMTRF supported this as well as CMTA, and we decided to scale the dose and try the same experiment in a monkey and ask, can we deliver an AV vector using lumbar intrathecal delivery to a monkey and have it reach the distal nerves and have engagement with PMP22 and knock it down? So what I'm going to show you in the next few slides is, are we able to see the virus going to the right places in the nerve? And are we getting knocked down at PMP22? Also, we had a whole safety study done. I'm not going to show you the safety data. I'll just tell you that we didn't have any adverse events in the monkeys. Okay, so here's the study design. Just making sure I have enough time here. So these are the doses that we used in mice. We consider these to be our safe doses in mice, safe and efficacious doses. And this is the doses that we used in the monkeys. We had 2 different dose cohorts, 4 animals each, and 2 different time points, 6 weeks and 12 weeks. And then we had 2 saline controls for each cohort. And then we had equal males and females. And the monkeys were delivered our product, AAV9, what we call our MIR871, which is the microRNA that knocks down PMP22, driven by a U6 promoter, intrathecally, and then we did all these analyses that I'm listing here. And this just demonstrates how we assessed biodistribution first. So this is intended to be a nerve. And so here's the proximal end, here's the distal end, and there's the joint. And so we sampled a number of different components of the nerve for different analyses, including DNA to assess where the virus actually went, RNA to demonstrate that the product, the microRNA that we've created, is actually expressed in those spots, and that it's knocking down PMP22 at the transcript level and also at the protein level. And so we were really happy and surprised to find that at 6 weeks and 12 weeks, we were seeing AAV genomes, so the DNA that we delivered in the distal part of the nerve in all the monkeys, except for the saline controls. The vectors present in all the nerves sampled, which are listed here. We did see a bit of a decrease in average genome copies over time, up to 12 weeks, but it never cleared. And we had relatively consistent expression of our microRNA up to this 12-week time point in all the monkeys. So the question then became, okay, well, the virus went to the right place. That's great. The microRNA is expressed in the right place. That's great. Are we seeing knockdown of the intended target, PMP22? Now, I want to remind you here that these monkeys are normal monkeys. They don't have elevated levels of PMP22, right? So we're starting from 100% and going below that. And we found in every case, roughly 30 to, well, here's our lowest here was 16% at the transcript level, but we're at the protein level, we're seeing about 30 to 50%, or maybe 30 almost to 60% in some nerves. Average knockdown of PMP22 quantified by Western blots. And you can see all those here. Here's from the proximal part of the sciatic nerve. Here's the distal part of the sciatic nerve. So again, we were very happy to see this correlation between where the vector went, where the microRNA is expressed, and subsequent knockdown of PMP22 in a monkey. Now, if we do the math on this, and this isn't intended to suggest that we can go, we don't really know how low we can go here, below normal levels, and still have healthy nerve. But I just want to remind you that the knockdown percentages that we're seeing in the monkey were about, let's say, in one nerve, 38% to 56%. If we start at 150% and just do the math, that means that we're normalizing PMP22 levels to what should be a healthy range. We're not going below normal levels of PMP22. So we think that there's very low risk of HNPP occurring. And we didn't see, like I mentioned at the beginning, we didn't see any negative in-life issues. We also didn't see, and here's just a couple of examples. We didn't see any changes in myelin structure. We didn't see any changes in functional performance of the nerve. So this seems to be a safe dose. And one of the things about AAV delivery and gene therapy that has been seen consistently in clinical trials is elevation of liver enzymes. So there's immunosuppression protocols now to deal with that, but we also noted that we aren't seeing any elevation of liver enzymes either, despite the fact that the vector does leach out of the spinal column and gets into the systemic body and actually transduces the liver pretty highly. And then finally, another big question we had and others have posed to us, I think it's a good question, is Cleopas has done some nice work demonstrating that AAV9 delivered in a mouse intrathecally hits maybe roughly 50% of the Schwann cells. Not all of them are transduced. So if we're trying to knock down PMP22 and we're trying to say, using a Western blot, that, okay, we're getting 50% knockdown, but we're only hitting 50% of the cells, does that mean that maybe some cells are getting over-silenced too much and some other cells aren't getting silenced at all? And that sort of represents an average. So the way we tried to address that question is to use this RNA-scope assay. This was something we did in partnership with ACD Bio. This is a company that's developed this RNA-scope assay. And what we asked them to do is create probes that could detect the microRNA and PMP22 and then co-stain cells, and this is done in a mouse, looking for consistency of silencing. So in other words, are we seeing microRNAs and PMP22 levels reduced in the same cells? And we were. There's a correlation between the amount of microRNA that's present in the Schwann cells and the presence of PMP22 levels or their knockdown. So we think, again, this is an exciting finding that demonstrates to us that what the knockdown we're seeing is consistent. So if the virus is getting into the Schwann cells, we're reducing PMP22 to levels that we think should be efficacious. So to summarize, in this study, we provide the first detailed biodistribution analysis of an intrathecally delivered AAV9 vector targeting Schwann cells. Other people have done this in monkeys looking at axonal targets, but we didn't know that you could hit Schwann cells very efficiently, so that's why we did this study. And we were, like I said, very pleasantly surprised at how well the data worked out. We've seen this stable therapeutic product that we call it MIR-871 up to 12 weeks. We've demonstrated target engagement and knockdown of PMP22 up to that time. That was our latest time point. And we didn't see any negative effects. I'll point out that we also did, Cleopas alluded to it earlier, we had a pre-IND meeting last year with the FDA, and we asked them about dosing, and there were actually a lot of questions, but one of the questions was, is our dosing scalable? The doses that we probably plan on using are going to be about twice what Dr. Boneman reported for the GAN trial. So they agreed that our dosing plan was good. and they gave us a blessing. So we're excited to try to move this forward now. And I think I will end. Yeah, this is just an extra slide in case I got a question. So I'm done now. Thank you for your attention.
Thank you very much. I guess we can have one question also here. Anything burning, or we can wait until the discussion at the end of the session? Right there.
Hello. I have a question about like long-term dosage. So when you showed your figures comparing your 12 weeks versus the 6 weeks on the non-human primates, your low dose in particular, I noticed that there was a decrease in the amount of the I forget what you were measuring, if it was the virus or the microRNA and that, but it had gone down from like 50-something% to 11. Would that suggest that you need to do continual dosage because there's a fading over time?
We don't know that for certain yet. What we're seeing up to 12 weeks is in almost all nerves. There was one nerve that we did see some loss there, but we're still finding the vector there. We're still finding the microRNA there. To answer that question, we have to do longer term studies. We just haven't done it. It doesn't, because we haven't seen any toxicity. I don't expect that we are having the vector cleared. The other thing I'll point out is that the payload does not encode a protein, so the payload itself should be non-immunogenic because the microRNA is just an RNA. So I'd be surprised if there was some sort of, toxic effect that was causing us to lose our vector, but I don't know if it's just not stable in Schwann cells for a longer period of time.
Okay, thank you. We have to move on.